Welcome, and thank you for watching this presentation on genetically modified organisms. My name is Christy,
And I'm Jacob.
and we are students at Truman State University.
To begin, let's define what genetically modified organisms, or GMOs, actually are. Genetically modified organisms are organisms, such as plants, animals, or bacteria, that have had their genetic information purposely altered by technology rather than through unguided natural processes. There are a number of methods that scientists can employ to genetically modify an organism. Rather than create an artificial process from scratch to insert genes into the genome of an organism, scientists instead utilize natural biological processes that are already suited to this purpose. One such method is to use bacterial plasmids to insert DNA into a cell. Some bacteria contain molecules called plasmids. Plasmids contain DNA that are independent of the rest of the bacteria's genetic material. Since we will be discussing GMO plants a lot in this video, I thought it would be good to share a video about how TI plasmids
Agrobacterium tumefaciens carries
are used to insert DNA into plant cells.
the plasmid, Ti, that can be transferred to broadleaf plants. The Ti plasmid is a useful vector for the genetic manipulation of plants. The plasmid carries a segment of DNA called tDNA, which integrates into the plant DNA. The tDNA carries genes that normally lead to tumor formation in the plant. However, researchers have succeeded in disrupting the tumor-causing genes and inserting new genes for resistance to disease, frost, and herbicides. The Ti plasmid can be removed from Agrobacterium, cut open with a restriction endonuclease, and mixed with DNA from another source, foreign DNA, that has been digested with the same restriction enzyme. The foreign DNA is then inserted into the plasmid. The plasmid can then be put back into Agrobacterium by transformation. Agrobacterium attaches to the plant cell, and a copy of the plasmid is transferred into the plant cell. The tDNA carrying useful information then integrates into the plant DNA. When the plant cell divides, each daughter cell receives the new gene. In some cases, the entire plant with the new trait can be generated from a single cell.
Another biological tool used by scientists to modify an organism's DNA is a viral vector. By nature, viruses work by invading cells and inserting their own DNA into the nucleus. This is how they reprogram a cell so that the cell will help them to reproduce. Scientists are able to take advantage of this process by removing the viral DNA and replacing it with DNA that they desire to be inserted into the cell. Then the virus invades the cell normally, but the DNA that is used to reprogram the cell imparts the characteristic desired by the scientist rather than propagate the virus. Now that its DNA has been altered, that cell and all cells that it gives rise to through normal cell division will now possess the desired trait.
Now we will talk a bit about who creates GMOs. This is a short list of some of the commonly known private companies that are developing genetically engineered crops. These 6 companies are the major players in the private sector. Many of them have extensive plant breeding operations across the world, and then add transgenic traits to the varieties they breed. Monsanto, based in Missouri, has the largest market share of GE crops in the world. Pioneer International, Syngenta, Dow AgroSciences, BASF, and Bayer Crop Science are others. These companies produce products such as corn, cotton, and soybeans that are genetically modified. There are a lot more companies working on genetic engineering than just the big 6. Many of them study individual traits, and if successful in their research, may license their technology to larger companies that can incorporate it into their crops. Some are developing their own varieties and breeds and seeking regulatory approval. Arborgen studies the genetics of trees, from wood quality to restoring endangered tree species such as the American chestnut. Florigene, based in Australia, focuses on introducing new colors into ornamental flowers and has produced the world's first purple carnations and the first true blue rose. Genetic engineering in agriculture is not exclusively done by private companies. Many public organizations and nonprofits are also developing the technology. Researchers that are not for profit include individual scientists, universities, government institutions, nonprofit organizations, and public-private partnerships. The golden rice that you will hear about later was a project started by individual scientists. Cornell University and the University of Hawaii teamed up to engineer a species of papaya, rainbow papaya, that can resist the dreaded papaya ring spot virus. As you can see in this picture, the center plants are flourishing. Those are the new species, while the unengineered plants are suffering from the virus around it. Another research source, the public-private partnership, is where the public and private sectors partner with each other in order to combine their resources to develop and take new and innovative biotech crops to market. One successful public-private partnership is the creation of water-efficient maize for Africa. Their goal is to develop drought-tolerant and insect-protected corn with a goal to make these varieties available to smallholder farmers in sub-Saharan Africa through African seed companies.
There are several motivating factors behind the creation of GMOs. One is to help nourish humanity. A great example of this is the Golden Rice Project. This golden rice is a variety produced through genetic engineering to biosynthesize beta-carotene, a precursor of vitamin A, in the edible parts of rice. This is meant to help alleviate vitamin A deficiency, which is estimated to kill 670,000 children under the age of 5 each year. Efforts are also being made to produce crops that have a greater yield. Flood-tolerant rice is one project that has helped farmers in South Asia not lose their rice during times of extreme rain. The drought-resistant corn spoken of earlier also fits this goal of producing greater yield. Generating profit is also an obvious goal of many companies who create GMOs. Selling seeds to farmers that are chemical resistant, such as Roundup-ready corn sold by Monsanto, can generate farmers money by helping their crops resist herbicides. This makes farmers satisfied and also makes Monsanto money. Another reason to modify is for medical purposes. One project in this realm is the creation of insulin-producing bacteria. This can help provide insulin to those who cannot produce it themselves, such as those with diabetes.
Now, why do some argue not to genetically modify organisms? Groups that oppose GMOs include anti-GMO organizations, political groups, and activists, a large amount of them based in Europe. Some of the primary arguments of this viewpoint are that GMOs are not safe for consumption. Studies have been conducted that conclude negative health effects from GMO consumption. GMO safety researcher Dr. Arpad Putzai was called to speak before Parliament and went public with research results that showed rats developing precancerous cell growth, smaller brains, and damaged immune systems. He was later fired and told not to speak about his research. This type of whistleblowing toward GMOs has happened numerous times and is usually shut down by the scientific community. Some see this as political power misused, and some as scientific research misconducted. Others are worried about GMOs because they think they are not tested properly. They argue that the testing is done by the very companies that stand to profit and are only conducted for short periods of time. This lack of long-term study makes sense since GMOs have not been around more than 35 years. This viewpoint sees this as a reason to be skeptical. Some believe most GMOs are not as good as they are advertised. See this video of anti-GMO activist Rachel Parent telling her opinion of golden rice.
Actually, funny you mention this. Golden rice was scrapped because it didn't work. And in order for the average 11-year-old boy to get enough vitamin A from rice, he'd have to eat 27 bowls of rice per day. And another thing is, the reason there's blindness isn't because there's a lack of vitamin A in the rice. It's because their diets are simply rice.
Another argument is that GMOs are harmful to the environment. One example of this is the GM salmon that was created to grow to full size in 16 to 18 months instead of 3 years. These salmon are only grown in land-based tanks because putting them in the ocean could harm the ecosystem where the salmon would have a survival advantage due to their growth rate.
On November 7th, a New York Times article was published explaining that the U.S. Department of Agriculture had just approved a genetically modified potato for commercial planting. Some of the reported benefits of this potato are decreased levels of acrylamide, which is a carcinogen or cancer-causing agent that is produced when potatoes are fried. Compared to unmodified potatoes, these GM potatoes produce 50 to 75% less acrylamide when fried. Additionally, the potato is more resistant to bruising during transportation. This is meant to cut down on the 30% of harvested food that never reaches the table for failing to meet purely cosmetic criteria. The potato is produced by the J.R. Simplot Company, which is based out of Boise, Idaho, the land of the potato. J.R. Simplot was the initial supplier of potatoes to McDonald's during the 1960s. The company is hoping that this product will assuage some of the public discomfort with GMOs and is calling it the innate potato because it does not contain genes from other organisms. Instead, it uses aspects of the potato genome to silence unwanted genetic factors.
After gathering all of this information, Jake and I feel that determining the validity of GMOs on a case-by-case basis is the best approach. Each organism is extremely different, and GMOs are created for varying purposes. Therefore, creating an overarching conclusion about GMOs is difficult. To conclude, each GMO, like salmon or rice, can be named harmful or helpful based on extensive specified research.
Thank you for watching!